I come across one American scientist near San Francisco who is using a yeast, an astonishing unicellular fungus, to produce a molecule to combat malaria. I head for the west coast of the USA. A yeast to make bread or brew beer. And as it happens, my appointment isn't at a laboratory, but in a microbrewery.
Hello. Hi. Welcome.
My name is Jay Kiesling. I'm a professor at the University of California, Berkeley. I'm also CEO of the Joint Bioenergy Institute in Emeryville, California. My definition of synthetic biology is the engineering of biology, manipulation of biological systems to accomplish some goal.
Jay Kiesling is one of the stars of this new discipline. As he describes the beer manufacturing process step by step, Jay tells me that his reputation came from his work on artemisinin, a molecule discovered in the 1970s to combat malaria. Until now, this molecule was extracted from a small, rare shrub, which only grows in a few parts of the world. Jay Kiesling has found another way to produce it by modifying the genome of a yeast.
We've taken the genes out of the plant that naturally produces artemisinin and put them into yeast. And so it's just like brewing beer. You put the yeast in a vat with water and sugar and some other nutrients, and the yeast grows up, and rather than spitting out ethanol, as it would making beer and wine, it spits out artemisinic acid, a precursor to artemisinin. And then we use a process, and it transforms from artemisinic acid into artemisinin.
Fortunately, what Jay gives me to drink is only beer. It all seems so simple. Making a new yeast and investing it with a role that nature had never thought of. It makes my head spin. Or maybe it's the beer. I don't think it was drinking beer that gave Jay the idea in the first place. A short drive away is the University of California campus at Berkeley, where he started out at a time when synthetic biology didn't yet exist.
I was fascinated with genetics. This is why I got into biology and why I took an engineering route. And so when I started as an assistant professor, I realized that we didn't have tools available to readily engineer metabolism inside cells. And so I started projects with my students on developing tools for metabolic engineering. But you can't just develop tools and not use them for something and not prove that they're going to be useful. And I decided, I should better think about some applications for these tools. So we started looking for applications, and we found artemisinin. I knew about malaria from a microbiology course in college. Knew nothing about artemisinin, and we looked into it and we said, boy, I think we can produce this, or at least a precursor to this. At the end of 2004, we had a $42 million grant from the Bill and Melinda Gates Foundation to finish off the process, to engineer a microbe to produce the product and then license it out to a company. Everything moved, in retrospect, pretty quickly. When you're in the process, you think, can't we move this faster? Can't we move this faster? There's people dying. Now, there's a large-scale process for producing it. They've produced on the order of 60 tons of the material, which is enough for 120 million people.
The wager paid off. Engineering life as one would engineer a machine. To understand how this is possible, we have to look at the basic tools of synthetic biology, using and combining the functions coded within DNA. Jay Kiesling first of all reminds me that each organism, each living cell, has one or more functions. Certain cells aid in digestion, Others convert light into energy for plants, or transform sugar into alcohol, like yeast, a surprising little unicellular fungus. But how do these cells know the nature of the work they have to accomplish? They quite simply have a manual or a program inscribed in their nucleus, their genome. The genome is an organism's instruction book for life. It describes all of a life form's functions. The book is made up of several chapters, which we call chromosomes. Chromosomes themselves are composed of genes. They form the sentences in the book. Each gene or each sentence is read like an instruction. All the genes together tell the cell what it has to do. All of this is written using an alphabet, DNA, made up of the well-known molecules A, G, T, and C, for adenine, guanine, thymine, and cytosine, which are always organized in pairs, the A with the T and the C with the G, known as base pairs. Genes are transmitted from one generation to the next identically, or are modified to respond to essential new functions or new requirements for the cell to survive. This is what we call evolution. If we know how to modify them and put them together, we can then change very simple functions into more complex ones. We can then imagine useful combinations that nature never attempted through evolution. With artemisinin, Jay has demonstrated that it was feasible and workable on an industrial scale. He transformed a cell into a chemical factory that produces active molecules. This concept enabled synthetic biology to avoid the problems that previously limited production capacities.
There are many advantages of producing a drug through microbial fermentations rather than having to extract it from a plant. One of the challenges with the plant is that it takes a significant amount of time between the time that farmers get seeds and companies decide how much they really want and contract with those farmers. The farmers get the seeds, plant them, harvest it, get the molecule or the extraction to the pharmaceutical company, and then they derivatize it and put it into the drugs. That process takes somewhere around 14 months to 2 years. And because of those long time scales, there's been large swings in the price and availability of artemisinin. It swung anywhere from about $200 a kilogram to over $1,100 a kilogram. And what that means is that when the price is high, people in the developing world can't afford it. When the price is low, farmers don't want to plant it because they can't make a profit, so they switch to other crops.
Be it Jay Kiesling or myself, or a developing world farmer, we're all the same. Whatever the invention, however avant-garde it may be, we also want it to be profitable. Jay's not only a scientist, he's also a businessman. He set up a company and runs another, J-Bay, a laboratory co-funded by various companies and the American government. Along the way, has he forgotten his noble ideas about saving the world?
I believe strongly in a well-regulated capitalistic system, but we have this system that gets products out to people. The company won't make any profit off this anti-malarial drug. In fact, and the University of California, which holds the patents to my work, gave the process away for free. We'll make a profit on something else that doesn't have to do with the developing world. I don't think that the non-profit model works for everything, right? What the company gets out of it is a well-honed organism. It produces artemisinic acid, but you can make a few changes in its genetic makeup now and it'll make many other things. And this is really a core demonstration of the power of synthetic biology. You build a platform and it can do many things. It's just like a computer. You can use a computer to write a document, a book, to give a presentation to an audience. You can use it to control the heating system in a building so that it's optimized. You can also use it to control some of the circuits in a car. Computers have many uses, just like some of these platform organisms will have many uses.
Jay Kiesling intends to make money from other applications derived from the same platform that's used to make artemisinin.
It turns out that I can apply that to many things. To producing drugs, to producing flavors and fragrances for ice cream, and for perfumes. I can apply it to producing fuels that would be carbon neutral, to producing the precursors to carpet that is produced in an environmentally friendly way that maybe has different characteristics. So my science is extremely narrow, but I can apply it to many different things.
Listening to Jay Kiesling, I wonder what the future of synthetic biology holds. Will it be the eagerly anticipated new industrial revolution? Some say there's no doubt.
We've got a long way to go. I would say we're still in the very early stages of what we'll eventually be able to do with synthetic biology. Right now, we use microbes that have been demonstrated by industry to be scalable, right? We can build chromosomes, and people can claim that it's a new life form, but for the most part, they're copying what nature gave us and making some changes in the DNA. Wholesale re-engineering of an organism, that's still many years off. I envision a day when, if you want to produce a particular product, you might custom build a cell for it. I'm hoping that we'll have plants in the environment that can help clean up the environment. Maybe that will sequester carbon from the atmosphere better, or that will degrade contaminants that have been accidentally spilled in the environment.
Meanwhile, one of the key applications of Kiesling's yeast concerns one of the planet's major preoccupations, ensuring energy supplies, and in a cleaner manner. As Jay confirms, few molecules offer an energy-volume-simplicity ratio as effective as oil. We therefore have to find alternatives, but which pollute less. He believes synthetic biology has the potential for putting an end to an economy based on non-renewable resources to one based on inexhaustible resources. Can we not imagine a living organism producing something that nature would never have imagined? Fuel, for example, produced directly by providing it with sugar or some other food. There'd be no need to dig, pump, and refine, just to draw it from a tap, like with draft beer. Well, this already exists, and one of the numerous laboratories working on the question is just downstairs from Jay's in the same building in Emeryville. I have a meeting there with Joel Cherry, their research director.
I'm Joel Cherry. I'm the president of research at Amherst here in Emeryville, California. We're a bioscience company that engineers microbes to produce hydrocarbons, primarily. The two that are currently on the market is a diesel fuel called farnesene and an emollient called squalane, which is a dimer of farnesene. If you smell farnesene, you would recognize the smell, but you probably wouldn't be able to guess where it came from. It's produced in nature from a wide variety of plants, and it's particularly concentrated in the skins of green apple. If you walk through our labs, when we have lots of fermenters running, producing farnesene. You get a slight smell of green apple.
It looks like this. It's the fermentation broth.
So that's farnesene.
It should smell like green apple skin.
Okay.
Farnesene is promising, but trying to produce it from apple skin would be ludicrous. Obtaining just 1 liter would require about 100,000 apples. This is where synthetic biology is a game changer. Jay Kiesling was a little evasive about how he transforms his yeast, so I asked Joel Cherry to tell me more about the techniques that are used to modify a cell's metabolism and get it to produce something other than what its genome originally planned.
Converting a yeast strain into a farnesene-producing strain really only requires the insertion of one new gene that encodes one enzyme that makes farnesene. We do that by reprogramming the yeast. We change the typical route that they take sugar, which is to ethanol, and we reroute that production so that the carbon that the yeast eat, the sugar that they eat, gets converted into the products that we want. We rewrite the programming inside the yeast cell using DNA. We introduce DNA from either synthetic DNA or DNA from other organisms.
Enzymes, coding, genes, synthetic DNA. I'm not sure it's very clear. I didn't even know there was carbon in sugar. In order to illustrate the delicate construction work carried out on a nanometric scale, Joel Cherry decides to explain things on a more familiar scale. He leads me along the building's corridors.
It's the same pipes.
If you think of a cell as a very complicated building with a plumbing system, the incoming pipe is sugar. The pipes inside a building go all kinds of places. What we really are doing is trying to pinch off specific pipes in that building and direct the flow of that carbon, the sugar, into a specific place. What we're doing when we're pinching off these pipes and trying to maximize flow of the carbon from the sugar into farnesene is we're decreasing the amount of carbon that goes into the yeast cell for things that we don't think it really needs and increasing the flow of the carbon into farnesene. We're the plumbers, and we have a very complicated building where we have 50%, 70% idea of where all the pipes go. We don't see all the connections, and there's still rooms that we don't understand.
It's incredible. All the equipment, all the investment, all the people working on the production of the product in question, farnesene. And Joel Cherry, the plumber, admits that he doesn't know the half of what really goes on inside a cell. And yet, this doesn't stop him or his team from making constant progress and gradually increasing their production.
What we're talking about is really designing the product we want to make outside the cell using computer-assisted design tools. And then using a collection of parts that is all the DNA that's been sequenced or the instruction manual that's been read so far to construct outside of the cell what it would look like. And then combine those pieces using the tools of genetic engineering. So it's really transitioning from a very artisanal or craftsman approach to an industrial approach. And the net result is we end up with a system that uses the engineering cycle, which is design, build, test, and learn. And we run that cycle recursively over and over again, iteratively, to make minor improvements on the original design. In synthetic biology, we're applying standardized parts, processes, and tools so that we can automate many of the steps we take when we're engineering organisms, much like we use when we engineer and build a car. The aim of the research is to maximize production, and we want to maximize the efficiency by which yeast converts sugar into our final product. How much farnesene is made per gram of sugar fed. They can make more than twice their cell weight in a few hours.
These little vials contain the most effective yeasts his team has managed to perfect after continually enhancing the capacities of the modified organisms.
So this is what will go to Brazil.
Thanks to these yeasts, Joel Cherry is now ready to move from the lab to large-scale production. Brazil, I love it. Joel Cherry gives me an appointment thousands of miles from the San Francisco Bay in a small town in the heart of Sao Paulo province. We meet again in the middle of a vast sugarcane field, where there's a vague smell of apples in the air. This isn't so surprising, as this is the site of his fuel production plant. It's eminently practical, as the ravenous yeast he's brought from California will have all the food it needs to reproduce and get down to work.
See, it controls out there.
Because it's a growing organism, you just need a little bit, and you can grow it into a giant 200,000 liter tank in Brazil, and it makes product. We have a plant with a capacity of about 40 million liters per year. It is built right next door to a giant sugar mill. And between our plant and the sugar mill runs a big pipe. And that pipe carries sugarcane, which gets fed directly into a giant steel tank that is called a fermenter. All we do is add our yeast cells to that fermenter with a few vitamins and allow it to grow. And it grows for a couple of weeks. It's constantly being fed sugar, and we're constantly removing product from the tank. While that fermenter is growing, we have 3 layers form inside the fermenter. The bottom would be the solid yeast cells. The middle would be the sugar juice that doesn't have any sugar anymore, so it's mostly water. And then the top layer is the oil. We can separate first the cells and then the water from the oil. And we end up with a product that's about 95% one molecule farnesene that could be distilled into a product that's about 98, 99% pure. So it's a really simple process.
But will the fuel produced here be able to compete with and later replace oil? Not really, not right away. Oil is still much cheaper than farnesene, 7 times cheaper, and so those who benefit from fossil fuels still have much to look forward to. Today, only a few buses fueled by farnesene run in the streets of Sao Paulo. But this won't always be so. The day oil prices go up, energy companies will turn to biofuels in a hurry. They're well aware of this, and so they're all already investing in synthetic biology. For the moment, however, Joel Cherry's company is benefiting from opportunities in the cosmetics sector. Synthetic biology has a role to play here too, making what's rare and precious available to the masses. Cost effectiveness is not easy to attain today. And indeed, in economic terms, clear figures are also difficult to establish. No financial expert would risk making hazardous estimates concerning the global value of the synthetic biology market, for example. A company like Evolva in Europe has developed cells capable of producing the molecule of saffron, a highly prized product for thousands of years. Until now, obtaining 1 kilo of saffron required 200,000 crocuses. One simple fermentation tank can today produce the same quantity. The same company, Evolva, is also counting on resveratrol, the molecule in red wine that certain studies claim has beneficial effects on the longevity of mammals. However, in order for it to be effective, one would have to drink 6 bottles a day, which might have certain side effects on the longevity of the mammal in question. With synthetic biology, there's no need to consume so much to age more slowly. Such benefits, promises, and products see the light of day in a fermentation tank that could perhaps soon become a household appliance. For scientists, as well as students, the grail is the discovery of the right combination of genes, the right formula to design a cell that produces something rare and precious. That would mean hitting the jackpot. Synthetic biology envisages new ways of producing, alternative avenues for research off the beaten track, even at home or in a garage. As it happens, this is what Thomas Landrin is working on in a lab unlike any other called La Paillasse in Paris, France.
Hello,
Hello, Thomas.
Thomas Landrin. Welcome to La Paillasse.
How long have you been here?
Just under 2 months.
Just under 2 months.
It's all quite new. Just before, we were in a squat at Vitry-sur-Seine. So for us, it was a huge step forward. My name is Thomas Landrin. Until now, I've been a doctoral student in synthetic biology, and at the same time, I set up La Paillasse, which now takes up all my time. I've always been a bit of a rebel. I've always loved doing things myself, and at certain times, when I don't get enough freedom in my own professional environment, I tend to move away and do things myself. La Paillasse has ultimately been an opportunity to create my own laboratory to work on the projects I've wanted to develop. We have 2 rooms here. There's another one in the back. These 2 rooms will be used for major programs. La Paillasse is an open, civically minded community laboratory for biotechnologies, and more generally for all new technologies. The idea is to provide people with a space where they can experiment and meet people who spark new ideas concerning these new technologies. We've tried to create a place where people have freedom, somewhere where we can give people who have an idea the possibility to work on whatever they want, as they want, and with whomever they want. In the end, what we're creating here is the same sort of thing that happened in the 70s with the geeks and the hackers.
The myth of Silicon Valley garages where DIY engineers created hugely successful startups never ceases to fascinate. Having worked in institutional labs, Thomas Landrin is convinced that the same model can be applied to synthetic biology. He wants to introduce a breath of fresh air to the field. But I wonder, a lab handling chemical products requires rather different facilities than a computer engineer might need. There are specific tools and safety measures to respect. And yet, for Thomas, the best proof of such garage labs' potential is the project he's developed himself for a biological ink.
One of the flagship projects at La Paillasse today is a project that will enable everyone to grow their own ink biologically. It has to be said that inks are some of the most polluting products on the planet. Amongst other things, they're made of heavy metals and some pretty nasty solvents. The idea is, 1, to provide a biodegradable alternative to these inks, and 2, to come up with an alternative that could provide a degree of self-sufficiency. And so it was through a discussion I had with a designer, Mary-Sarah Adonis, that we imagined a pen that could produce its own ink. Instead of having a cartridge, there'd be a bioreactor with bacteria. The bacteria would produce pigments, and we could use the pigments to write with. In exchange, we'd feed the bacteria, and so we end up having to feed our own pens. Even if the idea was completely insane, we began with the idea of using a bacteria that's naturally present in our ecosystem and which could perform that role. After about a month studying the relevant scientific literature, I managed to find a candidate that's non-pathogenic, which grows easily and which produces a blue pigment in vast quantities. We simply fed it with fairly classic nutrients, sugar, a little nitrogen, phosphate, etc. The pigment isn't toxic, and above all, it's biodegradable. This means we could use it to feed another microorganism, which could then produce other things. In this way, synthetic biology will enable us to obtain very practical results, and relatively rapidly, I believe. We have here some matter that's very interesting, that people can grow at home. They just have to grow a small bacteria culture. It will be open source technology. That means people can share it. We want to prevent it from being taken over by the big multinationals that have the ability to flood the market. So we'll probably have to apply for a patent and then issue free licenses.
The overall philosophy seems fine. Garage labs like this one are springing up all over the world. Although one can wonder about the scale of the projects that are developed there. They're generally very small, but as synthetic biology uses pre-existing bricks, it's not so much the technology that's important, but the ideas. It's not impossible that an interesting idea could emerge from the hacker's shareware universe. But I'm still not necessarily reassured about the use of potentially dangerous products or organisms outside official control.
It's clear today that there's no real surveillance of what we could call the garages, meaning those places which are beyond regulatory measures and which are invisible to national regulatory bodies.
So should we be afraid?
Making a synthetic virus today is extremely difficult. It's not even about whether you have the equipment or the required knowledge, it's about expertise and experience. And there's a certain form of transparency which generally means that when someone sets off in the wrong direction, they're very rapidly steered back on track. The framework has to be respected. You don't work with pathogenic organisms. You need licenses for that. You need the right equipment for that. And it's within that context that we've established the first ethical code for biotechnologies. La Paillasse is probably one of the biggest biohacker spaces in the world today. I think we'll soon be joined by other communities like this around the world. I wouldn't be surprised to see 100 or so within the next 5 years. We're already in the process of setting up a network. For example, there are Paillasses in Bordeaux and Lyon, and also in Lausanne, and one is being created in Manila, in the Philippines.
Thomas Landrin clearly has a broader vision. And it's true that when we see the popular success of the meetings and events organized around synthetic biology, there's good reason to be ambitious. The reference is no doubt iGEM, an event organized each year in Boston by some of the discipline's key figures, all from the prestigious MIT. Thomas Landrin himself has attended, and on his advice, I decided to take a look. Since 2004, this competition has seen students from around the world inventing cells with innovative properties. They work with a library of biobricks, genes from sequenced genomes that each have a specific biological function. The components they invent subsequently integrate the same open source database.
We made another one with cyanobacterium, and we put them into the registry, and also...
There's something for everyone, from the most serious to the most bizarre project in an atmosphere of intense emulation. I happened to meet a rather unexpected character among the young scientists, an FBI agent. So, maybe my concerns about security are shared by others after all.
My name is Ed Yu. I'm a supervisory special agent from the FBI's Weapons of Mass Destruction Directorate. I work in the Biological Countermeasures Unit. The mission of our unit is to provide a prevention mission on looking at how do we protect the life sciences, how do we prevent, detect, or deter the development, acquisition of biological weapons. The FBI has been a sponsor of iGEM since 2009. We conduct outreach to the students here, and we actually host a biosecurity workshop every year for the students, and the theme is Safeguarding Science in the Future.
So that's your... what are those cards?
These are biosecurity outreach and education trading cards. It does a nice job of explaining the differences in the microbes.
Ed Yu's background illustrates the evolution of governmental preoccupations concerning science and security issues.
Before joining the FBI, my background is in biochemistry and molecular biology. So I was involved in human gene therapy research, and I also worked for a biotech company doing cancer research. Post 9/11, the FBI diversified their workforce. So they hired individuals like myself, computer science individuals, people with foreign language skills.
We have the FBI represented by Ed Yu.
We're living in a different world now. It forced the FBI to change. We can no longer be just reactive. So if a crime occurs, we go in. Now we have to be proactive. We prevent another event like 9/11 from ever happening again, not only in the US, but anywhere else in the world. That has now become our priority mission. And I'll talk a little bit. So we're starting with this generation that's up and coming. These are the future scientists, future CEOs, and future policymakers. And as we've always seen historically, with any innovation and scientific advances, policy is always playing catch up. We actually host our own conferences too. So my program, we actually have FBI-sponsored conferences too. We're instilling this awareness. And I guess the best way to characterize it is not on my watch, meaning that during my time, whether I'm a student or a full-time scientist or a business person, that now that I understand what the security concerns might be, what can I do about it now? Well, how can I actively prevent this potential misuse or abuse or exploitation and notify the right authorities? The experts who can identify what the new security challenges are going to be, it's going to be the students, because they're the ones that are actually doing the work. They're in the best position to then teach the FBI that based on what I'm doing and with my now security awareness and I do my assessment, here's a potential vulnerability that you should be aware of. We are going to be dependent on that partnership to determine if there was some incident. We're going to need their expertise to help us determine in our investigations, was this purely an accident?
Ed Yu has no intention of minimizing the risks. Furthermore, in view of the extent of potential applications for synthetic biology, it's becoming increasingly complicated to evaluate them. In any case, rather than being called in merely to put out a fire, the FBI is being proactive. And yet I doubt that the dependence on the collective conscience, the not on my watch premise, involving mutual surveillance among scientists regulated by a code of conduct, will suffice in allaying the widespread doubts linked to synthetic biology. Such actions also reveal the inability of government agencies to keep an eye on everything at once. Fears concerning the abuse or misuse of science are not limited to questions of security. A good many NGOs are attempting to raise public and political awareness about other issues too. To learn more, I've come to Montreal in Canada to meet one of their representatives, Jim Thomas.
My name is Jim Thomas. I work for an organization called the Etc. Group, where I'm a program director. And we track the impacts of new technologies on society, on the environment, on economies.
Jim Thomas observes, criticizes, and sometimes denounces the failings of technoscience and scientists. He has recently pointed his finger at nanotechnology and geoengineering, but his favorite target today is synthetic biology.
The natural world isn't a machine. The idea that you're going to be able to improve nature is remarkably naive. There's a complexity there that has to be understood. And that complexity is not just ecological complexity, it's also social complexity and economic complexity. What worries me is that the field of synthetic biology works on some very simplistic premises that may not even be right. Synthetic biologists in their laboratories working on very specific applications are looking narrowly. They're not looking at the complexity of a social situation, of the ecosystems into which their organisms are going to be released. And in fact, often when you talk with synthetic biologists, they'll sort of admit that they can't even begin to think about that complexity.
Jim Thomas is particularly concerned about the pretensions of bioengineers and their blinkeredness. He travels around the world trying to mobilize the public at large. He takes me to Tunisia, where he's taking part in the World Social Forum, where science is one of the issues to be discussed. The country is undergoing upheaval and attracts militants from around the world. After his first intervention at the University of Tunis, Jim Thomas takes me to the city's Medina. He wants to show me some examples of traditional production that synthetic biology intends to replace. In his view, it would mean the end for small-scale producers. Even the most useful product can be criticized, in his opinion. For example, he lambasts the idea of manufacturing the yeast that produces artemisinin, which would appear to be of great benefit to humanity.
The claim that this organism, by producing this important anti-malarial drug, would be a major boon for the fight against malaria, but we very soon discovered that artemisinin was already grown by hundreds of thousands of farmers worldwide. And it instantly raised the question, well, what happens to those farmers? If there had been a lack of artemisinin and it hadn't been possible to get it from farmers, then this might have been a good. But that wasn't the reality. The reality is there was enough artemisinin.
Far from high-tech labs and industrial installations, Jim Thomas is above all interested in human beings, in the producers who are the most at risk.
That really pointed towards some of the sort of large economic changes that could happen once you could begin making commercially important compounds through synthetic biology.
Another issue has also drawn his attention, the question of sugar. Sugar is the raw material for synthetic biology. It's used in many projects, and supply and demand constraints could lead to problems.
Basically, you have a system where you're feeding large amounts of sugar to these engineered organisms. And on a small scale, that might be no problem. But if you begin to talk about producing large amounts of products, such as biofuels, for example, or plastics, you're going to need a large amount of sugar. And the production of sugar, particularly sugar cane in Brazil, is associated with all sorts of environmental problems. It's associated with social problems, with land grabbing. It's destroying one of the most fragile regions of Brazil. So this is potentially a very damaging ecological trend. Increasingly, the synthetic biology industry is saying, okay, then we'll not take any material from the land, we'll go to the sea. We'll see if we can use seaweed, and we'll grow large seaweed farms off the coast of China and off the coast of Chile. But once again, this is going to have ecological implications. So it's not possible to produce large quantities of something using biology as a basis without disrupting biology on quite a wide scale.
I sometimes get the impression that Jim Thomas is against everything. Against technology, against big business. But this is no doubt his role, since a form of counterweight and opposition to the buzz and the general enthusiasm generated by synthetic biology is essential.
The techniques that are used in synthetic biology of creating new living systems are tremendously exciting and interesting. And I think they're very useful for better understanding the way in which life processes work. They have a lot of value within the laboratory for science and for improving our knowledge of the world. Where I get concerned is when those techniques are taken to the marketplace for producing industrial applications and without really understanding the implications. And so until society has a way to address those disruptions, we need to be exceedingly cautious.
Caution is good. And words can be powerful, but can they really change anything? I asked Jim Thomas about the possible solutions that he and his group have envisaged.
I think what would be the best outcome, where if the governments of the world said, wait a second, since this is going to be so powerful, because this is going to have such wide impacts, let's slow down, let's think carefully, let's put a bit of a pause on commercializing these technologies and putting them out into the environment. That would require strong international governance. It would require a willingness to listen beyond the companies and the narrow group of scientists who are controlling this debate. But I think it would be a very sane decision for the next 150 years.
Whatever the field of research, communication between scientists and ordinary citizens has to be developed. These synthetic biologists, in managing to evolve evolution to make what nature never intended or imagined, have initiated a technological revolution that no doubt goes far beyond anything the world has seen before. A revolution I believe it would be a shame to scupper. Does it not have a great deal to offer us, a great deal to teach us about life itself? It can help our society to progress, to consume less and more efficiently, to preserve the environment, to find new therapies to cope with diseases. But unfortunately, there will be errors, there will be accidents, there always have been. It would be pretentious to claim the opposite. Will we be able to anticipate them? Will we be ready to face them? Such questions merit further investigation. There is still so much more to discover.